Dear all, welcome to the ISRT Lecture of 2023. I'm Ellen Merete Hagen, and I'm just going to introduce this year's lecturer, Dr. Monica Sousa. Just to tell you a bit about the ISRT Lecture, which was initially introduced by Mr. Ellis, a former editor of Spinal Cord. It's the International Spinal Research Trust, which is the UK's leading charity funding research around the world to develop effective treatment for spinal cord injuries. Their mission is to harness the power of international science to deliver revolutionary treatments. They founded many experimental treatments which had groundbreaking changes to spinal cord injury and its effects, and they cover both basic and clinical trials. And they are doing topics within neuromodulation, translational tools, neuroplasticity, bladder, bowel, and sexual function. Dr. Monica Sousa, who's this year's lecturer, graduated in biochemistry. She held a PhD in biomedical science from the University of Porto. She worked at Columbia University as a postdoctoral fellow, and since 2008 she's been leading the nerve regeneration group at the University of Porto. She has a lot of achievements. I counted 105 publications and 17 funding. And her work is focused on understanding the cytoskeleton organization and dynamics during axon growth and regeneration, in the latter case using rodent models of spinal cord injury. And she has uncovered the first mammal that is capable of regenerating and recovering function of the complete spinal cord transaction, the spiny mouse. Hence, her title is Translating the Spiny Mouse, Defining the Roadmap that enables the axon regeneration in mammals. Monica, please, the floor is yours.
Thank you, Ellen, for the very kind introduction. I really feel humbled and honored to be delivering the ISRT lecture today. And I'll be telling you about the spiny mouse and the importance. I hope to convince you that really using model organisms, and in this case I'm going to present you a non-model organism, is really important for us to try to understand the basic biology of a spinal cord injury, and hoping to that knowledge will inform us in designing new treatments. Okay, so throughout my talk I might refer to the spiny mouse by its scientific name, which is Acomish, and then to the regular mouse that we use in the lab that does not regenerate as mush. Okay, and I'll try to guide you through our efforts in trying to translate our findings in the spiny mouse to the regular mouse to make it regeneration competent. So, okay, most of you are well aware of the Edwin Smith papyrus, and basically what's described in this Egyptian medical papyrus is in many ways what we still do today in clinical practice. So what this papyrus is telling us is that an injury to our central nervous system, spinal cord injury, is beyond medical intervention, right? And if you take any textbook, what is normally stated is that regeneration of the adult mammalian central nervous system does never occur. And actually, this is caused by two different aspects that we generally study in the field. First of all, you have a number of different extrinsic factors that will make the environment of a spinal cord injury highly inhibitory for axon regeneration. And this is, of course, linked to the formation of a glial and fibrotic scar. So mammals respond to injury with scarring, okay? And the other thing is that in the adult mammalian CNS, the axons have a lower intrinsic capacity to grow. Okay, so these cells, so the neuron in the CNS during development has a really amazing growth capacity, and this is lost in the adult central nervous system. And this is a component that is also very important in respect to promoting axon regeneration following injury. Okay, so this is what happens in mammals, but as you might know, there are many other models that, or vertebrate models that are used to try to figure out the biology of spinal cord injury, and in many of them, regeneration following spinal cord injury is really effective. This is the case of the zebrafish, and the zebrafish is capable of regenerating after a severe spinal cord lesion because it builds a specific glial bridge, which does not occur in mammals. So, and by doing this glial bridge, the axons will be able to make new synapses. You also have the axolotl, which is an amazing aquatic salamander. And what the axolotl does following a spinal cord lesion is that it builds a blastema, so an environment filled with cells that are undergoing division, and this blastema is going to be able to regenerate the spinal cord. Of course, that when we're using vertebrates that are non-mammalian vertebrates to study the biology of spinal cord injury, one of the questions that always arises is what's the translational relevance of these findings, right? So something like 6 years ago, I learned that there was this really peculiar animal, a mammal, which I had never heard about. And as I was telling you, it's called Acomis cairinus, or spiny mouse. And it's really peculiar, and it came to my attention because it's really hard to keep on regular animal facilities for reasons that I'm going to tell you in a couple of minutes. And the thing is that one of the biggest colonies in the world is held in Portugal. Okay, Portugal is a very small country, so despite of the fact that we work in the north and the colony is in the south, I mean, we thought that we had skills on evaluating regeneration following spinal cord injury, so we should use this mammal. And the reason for that is, first I'm going to tell you a bit of the biology of the spiny mouse. So these animals are found in deserts in Africa, Middle East, and Asia. They're really more closely related to gerbils than to the regular mouse that we normally use in the lab. They get their name spiny because they develop guard hairs. They have very special diets, so they do not, we do not feed them just with the regular animal shell that we have in the lab, so they need to be supplemented with seeds and fresh fruit. If you provide them with food ad libitum, they will develop spontaneously type 2 diabetes. They're very peculiar because they menstruate, and rodents normally do not do so. They have very, very small litters, which is a problem in terms of doing research. So just around 2 pups are born per litter. And their lifespan is quite long for rodents, so they live around 5 years. And why did we become interested in the spiny mouse? Because there were reports already in the literature showing that these animals were able of an amazing regeneration of a number of different tissues. For instance, if you take the skin, so their skin is very fragile. You need to handle them really with caution. And if you don't do that, and if they develop a large skinned wound, what is going to happen is that in around 2 months, the wound will be completely regenerated. It's not like the regeneration that you'd be expecting with fibrosis. It's really healing with the formation of new hair follicles, skin, sweat glands, and fur. Okay, and this had already also been reported in the spiny mouse here. So we normally label, or many of us working with animals in the lab, we label them by doing small piercings in the ear. And with a spiny mouse, as you can see in the lower panel, whenever you do such a piercing, in 2 months also it's going to be fully regenerated. So this was basically what was known when we got started, but today it's also really known that these animals are able to regenerate their muscle, their heart, and kidney. So the question that we raised at the time was whether the spiny mouse was also capable of regenerating the central nervous system, so whether this regenerative capacity was not limited to external tissues such as the skin and the ear. So we started by contacting our colleagues in the University of Algarve, and we've done very mild lesions, so dorsal column hemisections, and what happened was that 2 months after lesion, there was no scar visible macroscopically following these very mild lesions. And this was the first indication that really in these animals, probably regeneration after spinal cord lesion was also possible. So then we've started doing complete spinal cord transections of the spiny mouse, and I'm going to show you the results that we obtained in animals that have been analyzed 2 months after complete spinal cord transection. So the initial part of my talk will focus really on this group of animals that have been analyzed 2 months after spinal cord transection. Okay, so in the bottom panel, you can see a regular mouse with a complete spinal cord transection, and it drags its hind limbs as is expected, right? And in the lower panel is the spiny mouse with a complete transection of the spinal cord. And as you can see, it's not walking perfectly. It's really walking, so it's able to stand the weight of the body, and it's really a coordinated type of movement. Okay, and this is quantified below. And when we did these studies, these initial studies, the reviewer asked us to do a second lesion in the injured animals to ascertain whether following this second injury, the animals were still able to regenerate. And in fact, they were. Not to the same extent, but they really were. And this was really amazing because this is really the first mammal that is shown to be capable of spontaneous regeneration after a complete spinal cord transection. And this kind of function was not limited to the recovery of locomotor function, but also to the bladder function. So one of the issues for our young researchers and postdocs working with spinal cord injury in the lab is that these animals have to have their bladders voided at least 2 or 3 times a day manually, right? And this is not the case of the spiny mouse. So the bladder function is completely recovered starting at around 1 week post-injury, but at 3 weeks post-injury, all the animals have fully gained their ability to void their bladders without any type of support. Okay, so after that, we really wanted to figure out what's happening in the spinal cord of the spiny mouse that is so different from what's happening in the spinal cord of other mammals following injury. Okay, so we started by collecting the spinal cords, and in the left-hand side, you see the regular mouse spinal cord, and you see that there is a very disorganized scar tissue that is formed in the lesion site, right here, there. Okay, so this scar tissue does not respect the initial architecture of the tissue, whereas in the spiny mouse, what we found was completely different. So there was the formation of this tissue that now is respecting completely the original architecture of the spinal cord. Okay. So when we looked at fibrosis was very much decreased in the spinal cord of the spiny mouse. And there was also a reduction in astrocytosis. So apparently, this animal is able to heal without massive fibrosis and astrogliosis. Of course, that by gaining function, we were expecting that to find regenerating axons in the spinal cord of the spiny mouse following injury, and that's actually what happened. So in the upper panel, you'll see a complete transection in a regular mouse. There are no axons crossing the scar tissue, and here in the bottom panel you have the spiny mouse, and this tissue that is formed between the rostral and the caudal parts of the spinal cord is completely filled with axons, which is quantified here. So it was not that just axon regeneration was going on. Many of these axons were already myelinated, as you can see here, by an immunofluorescence using myelin basic protein in red. So you see that this particular axon at 2 months post-injury is already well myelinated, okay? And then we looked at a number of different spinal cord tracts, descending tracts and ascending tracts, to check whether for all these different axonal tracts there was regeneration going on. And in fact, there is. For instance, when we checked the cortical spinal tract, what we found was that as opposed to the regular mouse, where no regenerating axons can be found, in the spiny mouse, caudally to the lesion site, there were multiple axons regenerating, and these axons did not follow the initial pattern of the CST, so there was this arborization present throughout the caudal part of the spinal cord, but the spinal cord was again filled with CST axons. And the same thing was found for the motor rough spinal tract. No regenerating axons for the regular mouse. The spinal cord filled with the regenerating axons for the spiny mouse. And actually, this was really long-distance axon regeneration. So I'm showing you here that the spiny mouse has regenerating axons that go beyond 2.5 millimeters, which was really the end of the slide. Okay? So this is really showing us that in this animal, axon regeneration takes place, and we could see axon regeneration following complete spinal cord transection for all the tracts that we have evaluated. And not only axon regeneration took place, but these regenerating axons were capable of forming synapses and of propagating signal. So when we did electrophysiology on the injured spinal cords, this is what you see in a mouse following a complete spinal cord transection. So there are no compound action potentials being registered, whereas in the spiny mouse, there you can see already it does not have the same amplitude, but there is already the recording of compound action potentials following the complete transection. So this initial piece of data is really showing us that this particular animal is capable of long-distance axon regeneration following a complete spinal cord transection. Of course, that the next question was to try to figure out which are the mechanisms that are enabling this specific mammal to do so. And to try to start to identify the mechanisms underlying this capacity, we started by doing RNA-seq analysis of the injured spinal cord and to compare really the transcriptome of the spinal cord of the regular mouse with that of the spiny mouse. So as this is not a normal model organism, actually it's a non-model organism, we do not have the tools that have been developed for other model organisms. For instance, the genome of the ISCoS had not been sequenced and assembled at the time where we started these experiments. So we had to collaborate with a group that does genomics that was capable of reconstructing the transcriptome of the spinal cord of the spiny mouse. And this enabled us to see that 2 months following spinal cord injury, there were 2 major pathways that were really different between these regenerative and non-regenerative species. which are inflammation-related pathways, which I'm not going to cover today, and then carbohydrate biosynthesis. So what's carbohydrate biosynthesis? So what we found is that a number of different enzymes which are related to the glycosylation of extracellular matrix proteins are very differentially regulated in the spiny mouse following a complete spinal cord transection. Basically, keratin sulfate proteoglycans that are an extracellular matrix proteoglycan, they seem to be really upregulated. Their synthesis seems to be very, very much upregulated in the spiny mouse following spinal cord transection. And this is led by this enzyme, this specific enzyme, beta-3-GNT7, that I'm going to tell you about in the next couple of slides. Okay. There's already a lot known on the importance of extracellular matrix glycosaminoglycans in the settings of spinal cord injuries. In fact, there are a number of different proteoglycans, and chondroitin sulfate proteoglycan is very well known to be very highly inhibitory for axon regeneration to take place. And there are a number of preclinical trials directed at really diminishing the levels of deposition of chondroitin sulfate proteoglycans in the spinal cord as a way of enhancing axon regeneration. But what the spiny mouse is telling us is that you probably don't need to just have the levels of chondroitin sulfate going down. You really need to upregulate the levels of keratin sulfates in the spinal cord. Okay? And in fact, what we saw when we compared the injured ISCoS or spiny mouse spinal cord with that of the regular mouse is that whereas in the spiny mouse there is this massive deposition of keratins following spinal cord injury, this is completely absent in the regular mouse spinal cord. So we now know what is the cell type that is expressing this beta-3-GNT7 enzyme in the spiny mouse spinal cord following complete transection. So it's really oligodendrocytes, okay? And we also know that this enzyme peaks very early on following spinal cord transection. So already at 3 days post-injury, its levels are over 100-fold increased when compared to the expression in the regular mouse spinal cord, okay? We started by doing in vitro assays to see whether the overexpression of this specific enzyme is really sufficient to enhance axon growth, okay? And for that, we compared the growth of neurites and axons by mouse neurons, the regular mouse neurons plated on top of cells expressing just a mock construct, a control, or this regenerative enzyme that I am telling you about. And what we found out was really that neurons plated on top of beta-3-GNT7 are really capable of growing longer axons than neurons that are plated on top of cells that are not expressing this enzyme. So what we're doing now is trying to translate our findings to the non-regenerative mouse, the regular mouse that we use in the lab. So we are developing AAV viruses targeting specific oligodendrocytes, and this will be delivered acutely after spinal cord injury in the regular mouse to figure out whether the overexpression or whether by increasing the levels of keratin deposition, we will be able to, that will suffice to increase axon regeneration post-injury. Okay, so we still don't know the biology below or underlying the permissiveness of keratins to induce axon growth and regeneration. We know, what we know is that the keratin interactors, here are some examples. They do not present major changes in the regular mouse and in the spiny mouse. And to try to really figure out what is the mechanism by which the overexpression of this specific enzyme, which is really very important for axon regeneration in ISCoS, how is it working? We're doing also RNA-seq of regular mouse neurons exposed to beta-3-GNT7 expressing cells to try to figure out what is this triggering on the neuron itself. Okay, so what I've been telling you so far are mechanisms or a cell extrinsic mechanism related to the composition of the extracellular matrix in the spiny mouse that enables it to regenerate. But our current data is also pointing to the hypothesis that we should not neglect the neuron intrinsic properties of the spiny mouse neurons. Okay, and the reason for that is that when you grow a regular mouse neuron, and this is a dorsal root ganglion neuron, on top of a very inhibitory substrate such as aggrecan, it will not, so its growth will be severely compromised. And this does not happen with the spiny mouse. So it seems that spiny mouse neurons are also not able to sense inhibitory cues in the same manner as the regular mouse neurons do. Okay. So we thought that we should, to get the complete picture of what's really happening in the spiny mouse, we should extend our initial transcriptomics analysis by doing RNA-seq at different time points post-injury, going from 1 day to 8 weeks post-complete spinal cord transection, and that we are not going to limit ourselves to study the extracellular environment, and we also acquired data on the neuronal intrinsic properties of the spiny mouse neurons. Okay, so we're doing part of this work in collaboration with the group of Grégoire Courtine, which is really building the atlas of the transcriptome atlas of a number of species, including the spiny mouse. And what I'm going to show you now is really what happens in the spinal cord, a macroscopic view, when we compare our data of transcriptomics in the spiny mouse and in the regular mouse. So in the spiny mouse here on the left, what happens is that you have an initial peak of transcription and translation going on, and it's shut down really early on following injury, and it's completely sustained in the regular mouse. The same thing for mitosis, so cell division. There's initially, of course, cell division in the spiny mouse that goes down very, very early on and is sustained in the regular mouse. And for inflammation, exactly the same. So inflammation initially is very similar in both species. It seems to engage the same type of response, but whereas it is shut down in the spiny mouse at around 1 week post-injury, inflammation persists in the regular mouse. Fibrosis and extracellular remodeling, a very similar picture. So it proceeds using the same type of mechanisms, both in the spiny mouse and in the regular mouse, up to 1 week post-injury, and then it's completely downregulated, whereas it's completely sustained in time in the regular mouse. And whereas in the spiny mouse, we see that neuronal markers and synapse markers start to go up at 1 week post-injury, okay, and they have very robust levels throughout the remaining time of regeneration. This does never occur in the regular mouse, okay? So in the regular mouse, all synaptic and neuronal markers go down early on after injury, and they never come back up. So what this slide is telling us is that the spiny mouse is capable of orchestrating a transcriptional switch very early on post-injury. So this species seems to be building a similar inflammatory profile, a similar fibrotic profile very early on, but then it manages to really shut it down and proceed with a annealing program. Whereas in the regular mouse and in mammals, including humans, this does never occur. So, of course, what we are trying to do now is to try to figure out what is this transcriptional shift. And for that, we are doing ATAC-seq that will enable us to see very early on which are the genes that are activated in both species and compare them. So by doing just a transcription factor enrichment analysis, this is just corroborating the findings that I just shown you. So what happens early on in both species is really similar, so we see the same transcription factors coming up. Then in ISCoS, at 1 week post-injury, you already have the activation of factors related to wound healing, neuronal differentiation. In the regular mouse, just pro-inflammatory cytokines and type 1 interference going up. Then later on at 3 weeks in the spiny mouse, we have transcription factors related to neuronal differentiation and synaptic plasticity, whereas in the regular mouse, the inflammatory response is completely sustained. So what we're doing now is really, by ATAC-seq, really try to figure out what might be the master trigger for these changes that we see very early on. Okay, so despite of the fact that we're really interested in covering these transcriptional switch that differs in these two mammals and that probably allows one of them, which is very closely related to have regenerative ability. We are also interested in studying genes that are differentially expressed in the two species by over 10-fold. Okay, and this is the case of a number of genes involved in the formation of collagen fibrils that, as you can see here, I'm showing you two of them that are massively upregulated in the regular mouse. Some chemokines, and in this case, I'm showing you a chemokine that is specifically peaking in the spiny mouse and is never seen in the regular mouse. And also there are a number of genes that are known in the literature as regeneration-associated genes. And the fact is that these genes are probably not really RAGs, as they are markedly decreased in the regenerative species in the spiny mouse. We also know that we have other interesting candidates, for instance, apolipoproteins. These apolipoproteins are completely different in the two species, and the way the spinal cord cells are expressing them is completely different, and we're also interested in covering if these differences are relevant in the settings of injury. And to make a long story short, what I've shown you today is that really the spiny mouse is the first regenerating mammal. So I've shown you that this species is capable of building a spinal cord injury site, which is basically scar-free and amenable to axon regeneration. And what we really want to understand is how this is possible in a mammal. And we know that we're just really scratching the surface, but we're very much convinced that the spiny mouse will offer us very important clues on what are the mechanisms that need to be upregulated and downregulated in a mammal to allow it to be regeneration competent. Okay, this is our final aim is always translation, and using the spiny mouse for translation, I'm going to show is not really a very innovative idea. So in 2017, there was this movie, Rampage, where this gorilla George was modified in order to have a number of special different features, and the sound is not coming up, but George has the growth rate of a blue whale, the strength of a rhinoceros beetle, and the speed of a cheetah. And in the end, tissue regeneration of an African spiny mouse. Okay, so this is basically what we want to achieve by using these species. So I can't end without thanking the amazing team with whom I've worked throughout the years. So this work was initiated in my lab by a really talented student, Joana Rodrigues, and is now being followed up by Sandra Braz, Eduardo Sousa, and Gonçalo Castro. I've had very generous support from Portuguese charity, Santa Casa da Misericórdia, Wings for Life, the Morton Cure Paralysis Fund, and the AO Foundation. So this Spiny Mouse project wouldn't have been possible because it took us a bit away from our comfort zone if we had not the support of amazing colleagues in our institute that are dealing with translational neurology, neural networks, phenotypic evolution, and glycobiology. And so I've been learning a lot these days at these meetings. So you're not my usual audience, and I've been learning a lot from a more clinical-oriented audience. And we try to never lose focus of what our subjects are and of physiology. And to do that, we interact quite a lot with Maria Cunha, that is here in the audience, and she's leading the spinal cord injury unit in one of the major rehabilitation centers in Portugal, and with Esther Rosa, which leads a patient association that deals with spinal cord injury patients in Portugal. And thank you.
Excellent. Thank you so much. I think we all have learned a lot, and it was really interesting. I'm sure there are questions in the audience. Please raise a hand. And I get some exercise.
Hello, I'm Géraldine Jackman from Montreal, Canada. Thank you very much for your presentation. I actually hesitated to come and see another mouse study, but I don't regret coming here. My question is, have you had, with this mouse, have you had other experiences of other types of neurological injury, such as peripheral nerve injury and stroke or TBI? Thank you.
No, no. We're really very much focused on spinal cord injury, and probably our lab will not go beyond spinal cord injury, but this finding has raised immense awareness on the existence of this mammal that is capable of regenerating. So I'm sure that further studies with other injury models will appear.
So Norbert Weidner, Heidelberg, thank you very much for this fantastic overview about regarding your regenerative model. I was just wondering, so when you show regrowth of descending motor pathways, which is quite striking, but then in the dish you look for DRG neurons, right? And those are the ones that go up in the cord. So how does that look in the spinal cord? So meaning in the lemniscal tract, for example. So how do they regenerate?
They also. So I just shown you two descending tracts today, but we traced several different ones, and we traced the dorsal column tract, and it also regenerates. So that's why some of our follow-up studies are being done in DRG neurons, because they're much easier to isolate than other neuron types.
Thank you very much. I think after your presentation, we can see the light end of the tunnel. Hoping we'll see this progress in our lifetime, and really for us, for our group of patients. So we can see some light end of the tunnel. Thank you very much.
You're welcome.
I'd like to ask Simon Shaw from Stoke Mandeville, why are there not more of these little mice running around? Why are they only based in certain parts of the world? Because if they're so resilient and they do all these amazing things from wound healing to spines, why aren't there more of them?
I have no idea. They're really kind of alien, and they're really fond of deserts, so very dry and warm habitats. That's why you find them in deserts in Africa, Middle East. And and probably also that's why we have the largest colony in the south of Portugal. So it's not easy to breed these animals under normal conditions in facilities, but actually in the south of Portugal, they're not bred in a regular facility. They're almost in a shed.
In my pre-med, we often related anatomy and structure to function in different animals, embryos, all sorts of things. And I think there's a clue there, to try and find out, how what advantage does this give, apart from the obvious, in the environment, and yeah.
I think you're right. So in many of these papers, it's suggested that the spiny mouse evolved this regenerative ability. Initially, the skin was the major thing here, to evade the predation. But I think that all this data that is coming out, showing that different organs are able to do that in the spiny mouse, are really raising interesting questions, and we should not overlook what you've just suggested, yes.
Thank you very much. Alison Morland, Brunel University, London. In your videos we saw of the spiny mouse, when it was running around at 2 months, it wasn't walking normally. Did it ever regain the ability to walk with a normal gait pattern? And if not, do you any idea why?
The answer is that we've just followed animals up to 2 months post-injury. We don't have longer follow-ups. So I really don't know if we had followed them for larger periods of time, they would completely recover their function. When I refer to their locomotor pattern is not completely recovered because many of these animals hop, which is not the case of the regular mouse. And only a part of these animals regain their hopping capacity.
This is Harvinder from New Delhi. One of the main challenges that we have faced is in the translation, because there have been quite a few preclinical studies which have shown good success, but translation has not been possible. So do you anticipate the same problem here, or should we be more optimistic with this technology?
Okay, translation is not easy. so what I've shown you, the message needs to be optimistic, right? We now found a mammal that is able to do what no other mammal does, and we need to understand the biology underlying that, right? But this does not promise that translation will be effective. We expect that because now we're talking about mammals, but we need to check it.
Yeah, thank you. We tried to also replicate 2 of the clinical models which had proven to be successful elsewhere. One also in Portugal, and we published it also. Well-designed clinical trials have not been able to replicate it, and that's the trial. But that also, such preclinical models need to be looked into. So congratulations for your study.
Thank you.
Any more questions from the audience?
Hi. Thanks for the presentation. I'm Ram from Sheffield. Just going to follow on from the previous question, do you have any links with a clinical setup? Because often, having worked in the research field and in the clinical field, there are very few meetings where both these teams actually meet together and discuss areas of expertise to actually translate these things. So while it is great to see advances in the animal models, unless there is an opportunity to understand what happens in the clinical world and vice versa, I think translation will look very far away.
No, you're absolutely right, and so I think that we should strengthen the collaboration between both more clinical-oriented and more, let's say, academic-oriented research or preclinical models. Yes, we do interact with a number of neurologists that see spinal cord injury patients, some of them based in Portugal and some of them based abroad. but we're not yet at the point where we aim at translating to humans. We first need to prove that our data is relevant in a model organism that does not have regeneration competence, and that's why we're initially translating our findings to other rodents that normally do not regenerate, and only after that we might think of really engaging in a preclinical or clinical trial.
Excellent. Given the numbers of questions you have received, I think this was a highly successful lecture. So thank you so much.
You're welcome. It's been a pleasure.
And may I just remind people of the AGM here at 4:45? No, 4:45. Yeah. And then we have the conference dinner tonight. Please join us. And thank you very much.